Welcome, ladies and genomes. I'm Ethan. I'll be your tour guide on the path through the central dogma.
I'm Michaela. I'll be your camera guru and editor-in-chief.
Hi, my name is Aaliyah, and I'm your resident scriptwriter.
Today, we will be illustrating the process by which genetic information passes from deoxyribonucleic acid, DNA, to ribonucleic acid, RNA, and finally to the formation of fully folded polypeptides. The first step of the central dogma is DNA replication. Or the unzipping and cloning of the DNA molecule. DNA is the package that holds the genetic instructions used in growth, development, and reproduction in all living organisms. The molecule is composed of two complementary helices made of deoxyribose sugar and phosphate groups that hold nitrogenous bases known as nucleotides. The nucleotides pair exclusively adenine to thymine, cytosine to guanine, and join together using hydrogen bonds. DNA replication starts with the enzyme helicase unzipping the double helix to create two template strands, known as the leading and lagging strands. The area unzipped by DNA helicase is known as the replication bubble. The leading strand uses a singular RNA primer molecule to begin its replication. Then DNA polymerase I adds nucleotides in a continuous motion from 5' to 3' with the path of the replication fork. The lagging strand travels from 5' to 3' opposite to the replication bubble direction, away in a discontinuous manner. This synthesis requires many RNA primers to begin the adding of short DNA fragments called Okazaki fragments by DNA polymerase I. After the Okazaki fragments have been added, DNA polymerase I replaces the RNA primers with nucleotides and nicks the Okazaki fragments for ligating. DNA ligase then joins the fragments to form a complete copy of DNA on the lagging strand. Each template strand receives its complementary nucleotides to form two identical DNA copies. The second step in the central dogma, the first step in gene expression, is transcription. Transcription is when the genetic information in DNA is copied into mRNA to travel outside of the nuclear envelope for translation, but more on translation later. Transcription contains three steps of its own: initiation, elongation, and termination. These steps occur at stretches of DNA that code for singular RNA molecules, a transcription unit. Initiation begins with transcription factors recognizing the TATA box, a sequence of nucleotides, specifically TATAAA, that signals where transcription begins. RNA polymerase binds to the promoter, forms the RNA polymerase transcription complex, and separates the DNA strands. The elongation phase of transcription refers to the process through which nucleotides are added to the growing RNA chain. As the RNA polymerase moves down the DNA template strand, the open RNA-PT complex bubble moves also. The complex reads and matches complementary RNA nucleotides to the template strand of DNA in the 5' to 3' direction. This process is similar to DNA replication, with the exception that the nucleotide uracil pairs with adenine in place of thymine. In termination, RNA polymerase continues pairing nucleotides until it transcribes a terminator sequence, where the newly formed pre-mRNA is cleaved. Only the pre-mRNA will be processed and used in translation and is released as the complex continues transcribing. In protein-coding genes, RNA polymerase II continues transcribing for up to thousands of nucleotides until 5' exonuclease consumes the extraneous code and releases the RNA polymerase.
You have been terminated.
A modified 5' guanine cap as well as a 3' poly A tail are added to the pre-mRNA. These additions assist in the exportation from the nucleus, protect from enzyme degradation, and facilitate attachment to ribosomes. Following termination, pre-mRNA must go through a series of processes in order to create translatable mRNA. Collections of small nuclear ribonucleoproteins, or SNRPs, join together and form spliceosomes. At these spliceosomes, pre-mRNA is spliced, removing introns, or non-coding sequences, and then exons, coding sequences, are spliced together to produce translatable mRNA. The final step in the central dogma is translation. Translation is the building of polypeptides through interpretation of the code carried by mRNA. Amino acids are coded in sets of 3, known as triplets in DNA, codons in mRNA, and anticodons in tRNA. Translation takes place in the cytoplasm at the ribosome. Translation, like transcription, contains 3 substeps: initiation, elongation, and termination. mRNA enters the ribosome to bind with specific nucleotide anticodons carried in tRNA. tRNA is a molecule around 76 to 90 nucleotides long that carries amino acids to the ribosome. Initiation begins when the mRNA start codon, AUG, pairs with the tRNA molecule carrying the starting amino acid methionine. Following initiation, elongation is when tRNA anticodons continue to bind to codons in the A site, amino acyl site, of the ribosome. The amino acid will then be joined to the chain and the tRNA translocates to the P site. The P site, or peptidyl site, holds the tRNA that carries the polypeptide chain. After transferring the polypeptide chain to the new tRNA, the old tRNA in the P site translocates to the E site, or exit site, where it is held, now missing its amino acid, until it is released from the ribosome. Termination occurs when the polypeptide is extended until the mRNA stop codon is reached. The release factor bound to tRNA binds to the stop codon and the polypeptide is released from the ribosome.
Terminator is on the job.
Following translation, the polypeptides produced are short, unfolded amino acid sequences that, once stitched together and folded, will form a complete functioning protein. The amino acid sequence of the polypeptides causes the chain to bind at amino groups and carboxyl groups. These bindings are what gives the protein its structure and ultimate function. In conclusion, the central dogma is the description of vital processes that create the basis for life-sustaining functions. Gene expression is the system that utilizes life's universal code, DNA, to create functional gene products that work on a much larger scale to support life at all complexities. Thank you.